Hello everyone, welcome back to the Medi Queries. Today we are going to discuss a very important topic in the pharmaceutics, that is extraction processes. So imagine you are preparing a tea. So you place tea powder in hot water. After sometimes the color, flavor and the chemical constituents of the tea move into the water. So what happened here? A simple extraction. So in pharmacy, we use the same basic scientific idea to separate medicinally active constituents from the crude drugs. So by the end of this presentation, you will clearly understand the maceration, percolation, Soxhlet extraction, and the factors affecting the choice of extraction process. So keep one simple question in your mind. So how can we remove the useful constituent and leave the unwanted material behind? So let's begin. Definition of extraction. So extraction is the treatment of the plant or the animal tissue with a suitable solvent to separate the medicinally active constituents. For example, imagine a medicinal leaf. So the entire leaf is not usually required in a formulation. So we may need only a particular chemical constituents present inside its cells. When a suitable solvent contacts the drug, suitable constituents dissolve in the solvent. So the unwanted insoluble material remains behind. So a very simple example is coffee preparation. So hot water extracts the soluble color, flavor and other constituents from the coffee powder. So remember, drug plus suitable solvent leads to separation of the soluble constituents. So before learning the extraction methods, so we must understand 4 important terms. The first one is menstruum. So menstruum is the solvent used for the extraction. For example, water or alcohol may act as a menstruum. The second is mark. So mark is the insoluble residue remaining after the extraction. So think about tea waste remaining after preparing the tea. It is similar to mark. And third, percolate. So the liquid obtained during percolation is called the percolate. Finally, extraction means complete removal of the extractable constituents from the crude drug. So students, remember this simple sequence. Menstruum enters, constituents dissolve, percolate comes out, and the mark remains behind. Types of extraction processes. There are several extraction processes. We have infusion, decoction, maceration, percolation and digestion. So today our major focus is maceration and percolation. So maceration can be simple or multiple maceration. So percolation includes a simple percolation, reserve percolation, modified percolation and continuous hot extraction. So how do we select one method? So we cannot randomly choose an extraction process. So the nature of the drug, stability, solvent, cost and the therapeutic importance influences our choice. So we will understand these factors at the end. Maceration. So the word maceration can be remembered using one simple word, soaking. So in maceration, the crude drug is kept in contact with the menstruum for a specific period of time. So imagine soaking rice in water. So water slowly enters the rice here. Similarly, during maceration, the solvent penetrates the drug tissue. So soluble constituents dissolve first and move into the surrounding solvent. So the basic principle is diffusion and dissolution. So whenever you hear maceration, remember keep, soak, dissolve and separate. Simple maceration. So now let's understand simple maceration for organized drugs. First, take the drug and the whole quantity of the menstruum. Keep them together for approximately 7 days. So during this period, shake the container occasionally. So after the required time, stain the drug, that is liquid, press the mark and mix the liquids and finally filter. So why 7 days? Because extraction is not instantaneous. So the solvent needs a sufficient contact time to penetrate the tissues and dissolve the constituents. So it is like soaking tamarind in water. So if you remove it immediately, extract is poor. So give it time and more constituents enter the water. Why shaking during a maceration? So here is an interesting question. So why should we shake the container? Suppose the drug particles are surrounded by the solvent. So after some time, the solvent close to the drug becomes rich in dissolved constituents. So we call this a relatively saturated solvent layer. So if this layer remains undisturbed, so further diffusion becomes slower. So shaking removes this layer and brings a fresh menstruum in contact with the drug surface. So this improves the diffusion and increases the extraction. So imagine adding sugar to the tea. So if you don't stir, dissolution is slower. So when you stir, the fresh liquid contacts the sugar. So the same basic concept applies here. Okay. Pressing of mark. So after straining, some valuable liquid remains trapped inside the mark here. So should we throw the mark away immediately? No. So the mark is pressed to recover the retained liquid. So a filter press, a hydraulic press or a hand press may be used. So for small quantities, a manual squeezing may be sufficient here. So imagine squeezing a wet sponge. So even after removing the sponge from the water, it holds like some water. So when you press it, additional liquid comes out. So mark behaves in a similar way. Okay. So the purpose of pressing is to recover the valuable retained extract. Okay. And simple maceration examples. So examples of the preparation made using this maceration includes tincture of orange, tincture of lemon and tincture of squill. So look at orange peel. So it contains aromatic and the soluble constituents. So when a soluble menstruum contacts the material for a sufficient time, so these constituents are extracted. So the same general principle applies to the lemon and the squill preparations also. So whenever studying examples, don't simply memorize the names. So ask yourself, what is the solvent trying to remove from this crude drug? So the question makes the pharmacognosy and the pharmaceutics much easier. Okay. So maceration with adjustment. Maceration with adjustment is commonly used for unorganized drugs. So initially, about 4/5 of the menstruum is used and the drug is macerated for 2 to 7 days. So shake occasionally and filter the liquid. So now here is the important difference. A portion of the menstruum is passed through the filter. Finally, the volume is adjusted to the required quantity here. So why is it called the maceration with adjustment? Because the final volume is adjusted. So that is your memory trick. Okay. So why mark is not pressed? In maceration with adjustment, the mark may form in a compact gummy mass. So it does not retain the menstruum in the same manner as organized drug material. So therefore pressing is unnecessary. So the active constituents dissolve in the menstruum and the final volume is adjusted. So students often become confused here. So in simple maceration, we discussed pressing the mark. So here the mark is not pressed. So always connect the procedure with the nature of the crude drugs. Okay. Examples of the maceration with adjustment. So examples includes tincture of tolu, compound tincture of benzoin and tincture of myrrh. Tolu, benzoin and myrrh are resinous materials here. So this is why understanding the physical nature of the drug is extremely important. So if I give you a soft leaf and a sticky resin, can you expect both to behave identically during the extraction? So definitely not. So the extraction process must be adapted to the material. Okay. Multiple maceration. So now imagine washing a dirty cloth. So would you get better cleaning with one large wash or repeating washing using the fresh water? So repeating washing can improve removal. So this is the concept behind the multiple maceration. So the menstruum is divided into portions and the drug is extracted repeatedly. So fresh menstruum contact the drug again. So this improves the extraction efficiently here. So the two important types are double maceration and triple maceration. First we will look at double maceration. So in double maceration, the menstruum is divided into two portions. Okay, during the first maceration, the drug is kept with the first portion, usually for 48 hours. So then strain and press the mark. So now the mark is treated with the second portion of the menstruum. So macerate for approximately 24 hours here. Again strain and press. So why is the first period longer? So initially, the drug contains the maximum quantity of the extractable constituents and require the effective contact. So the second extraction helps recover the additional remaining constituents. Okay, that's why it is less. So final steps of the double maceration. So after both macerations, the liquids are mixed. So the combined liquid is allowed to stand for about 14 days and then filter. So examples includes concentrated infusion preparations. So the important idea is to separate the extraction stages are finally combined. Okay, so think of the squeezing juice from fruit twice and collecting both portions together. Okay, so the second extraction may look weaker, but it still contains useful constituents. So combining both improves overall recovery here. Okay. Triple maceration. Triple maceration divides the menstruum into three parts. So first maceration 1 hour, second maceration is for 1 hour, and third maceration is also for 1 hour. So after each maceration, stain the liquid. So after the final maceration, the mark may be lightly pressed. So why 3 extractions? Again question. So every fresh portion of the menstruum creates a new concentration difference. So this encourages the more constituents to move from the drug into the solvent here. Remember, fresh solvent creates a fresh driving force for extraction here. Okay. Final steps of the triple maceration. So after completing the triple maceration, the second and the third macerates are combined. So they may be evaporated to the specified extent. Then the first macerate is mixed. Okay, alcohol and water may be added as required. So the preparation is allowed to stand and finally filter. So notice how carefully the procedure is controlled. So extraction is not simply putting a plant into a bottle. So it involves solvent ratio, contact time, concentration, evaporation, and volume adjustments. Okay. So maceration versus infusion. So now let's compare both maceration and infusion. So maceration commonly uses an alcohol or aqueous solvents, and the extraction period may be 2 to 7 days. So it is generally performed at room temperatures only. So the mark may be pressed depending on the procedure. So whereas in case of infusion, commonly uses water, either cold or hot. So the preparation time is much shorter here, approximately 15 minutes. So think about preparing tea. So that is closer to infusion. So but soaking medicinal material for 7 days is closer to maceration. So the major differences are solvent, time, temperature, and procedure. Okay. Organized and unorganized drugs. So organized drugs possesses a definite cellular structure here. Examples includes leaves, roots, and barks. So unorganized drugs are the products such as gums, resins, and exudates. So for organized drugs, a simple maceration may be used. For unorganized drugs, a maceration with adjustment may be preferred here. So this is a very important exam concept. So remember, organized means structure, unorganized means no definite plant organ structure. So the nature of the material influences the extraction technique. Okay. What is percolation? So now we enter another major extraction process, that is percolation. Percolation is the slow passage of the menstruum through a packed column of drug. So imagine rainwater passing slowly through layers of soil. So water enters from the top and moves downwards. Similarly, menstruum passes through the packed crude drug here. As it moves, soluble constituents will dissolve. The liquid leaving the percolator is called the percolator. Simple flow, menstruum down column and percolate out. Types of percolation. So the major types are simple percolation, reserve percolation and modified percolation, and continuous hot percolation or Soxhlet extraction. So each method has a specific purpose. So simple percolation is the basic process here. So reserve percolation protects the stronger portion of the extract. So modified percolation aims to improve the efficiency here, and Soxhlet extraction repeatedly uses hot solvent. So don't memorize these as four isolated names. So think of them as a improvement of the basic extraction idea here. Okay. Types of percolators. So percolators may be conical, cylindrical or steam jacketed. So the conical shape reduces the chance of choking. A cylindrical percolator has an equal diameter and is suitable for certain materials here. A steam jacketed percolator uses the heat to increase the solvent action. The apparatus design matters because the solvent flow must be uniform. So if the solvent finds one easy pathway, it may bypass the other portions of the drug here. So this is called the channel formation. So good equipment design helps prevent inefficient extraction. Stages of simple percolation. So simple percolation has important stages. First, imbibition, second, packing, so then maceration, and finally percolation. So remember the sequence as moist, pack, wait and flow. So students repeat it once. So moist, pack, wait, flow. So if the powder drug is packed dry without proper preparation, swelling may occur inside the percolator and create some problems. Imbibition means moisturizing the powdered drug with menstruum before packing. So the moistened drug is generally allowed to stand for approximately 4 hours. So during this period, the tissue will swell. So entrapped air escapes and the material becomes suitable for uniform packing here. So imagine a dry sponge. When your water first touches it, air may remain trapped. So after complete wetting, the sponge absorbs the water uniformly. So the basic concept help us understand the imbibition here. Okay. Importance of imbibition. Why is imbibition so important? So it prevents the blockage. It allows swelling before packing, and it improves the uniform packing. So it prevents the formation of the impermeable layers, and it also removes the entrapped air also. So suppose if you take a dry drug is tightly packed and then solvent is added, so the drug may swells inside the percolator. So pressure and the blockage can help. So therefore imbibition is not an unnecessary extra step. It is a preventive step that improves the extraction here. Okay. Packing of percolator. So now observe the packing arrangement. From top to bottom, we have menstruum, washed sand, filter paper, drug, glass wool and outlet. So each layer has a role. So the drug is the extraction material, glass wool supports the drug bed, filter paper and washed sand help maintain the upper surface, So menstruum enters from the top and the percolate leaves through the outlet. So you can compare the percolator to a carefully designed filtration column. So the arrangement must support controlled solvent movement here. Packing procedure. First pass the drug through a suitable sieve. So then place the solvent moistened glass wool and the drug little by little. So press lightly after each addition. So pack the material to approximately 2 to 3 or 2/4 of the percolator capacity. So place the filter paper on the top. Finally add the washed sand. So why add drug gradually? So again another question. So because dumping the entire powder at once may produce uneven packings here. So uneven packing means uneven solvent flow. So uniform packing gives a better extraction here. Okay. So role of washed sand. So washed sand protects the top layer of the drug here. So when the menstruum is poured directly on the powdered drug, the surface may become disturbed. So channels can form. So the solvent may follow these channels instead of passing uniformly through the drug bed. Washed sand distributes the menstruum more uniformly here. So imagine the pouring water directly onto loose soil. So a hole may form. So but if the water is gently distributed, so penetration is more uniform here. So this is the role of the washed sand. So maceration in percolator. So after packing, so the sufficient menstruum is added. So the drug material becomes saturated here. So the outlet is closed when liquid begins to appear. So a layer of the menstruum is maintained above the drug here. So the percolator is closed and allowed to macerate often for 24 hours. So why wait? So because the solvent needs a time to penetrate drug cells and dissolve the constituents. So percolation includes a maceration stage before continuous collection begins. Okay. So percolation stays. So now the outlet is open. So menstruum flows slowly. So the percolate is collected. So at the same time, the menstruum is maintained above the drug column. So the recommended flow may be around 10 to 30 drops per minute depending on the procedure. So why should the flow be slow? So if the solvent rushes through the drug, contact time decreases. And imagine the washing a tea bag under rapidly flowing water for 1 second. So would you obtain the strong tea? No. So controlled contact improves the extraction here. Okay. Testing during exhaustion. So how do we know that the extraction is complete? We test for drug exhaustion. So special chemical tests may be used. So we can also observe the color of the percolate. So the absence of the characteristic color may indicate the reduced extractable constituents. So the absence of the bitter taste may also be useful for critical certain drugs. So but remember, in pharmaceutical practice, a specific analytical or the chemical tests are more reliable here. Okay. So extraction means that useful extractable constituents have been sufficiently removed. Again I'm telling, exhaustion means that useful extractable constituents have been successfully removed. Final steps of the percolation. So after percolation, the mark is removed. It may be pressed. So the pressed liquid is mixed with the percolate. So the volume is adjusted with the menstruum and finally filtered. So again, think about the wet sponge example. So the mark can retain the valuable liquid here. So pressing helps recover it. So the final volume must be accurately adjusted because the pharmaceutical preparations requires the consistency. So every batch should be controlled quality. Okay. So examples of the simple percolation. Examples includes tincture of belladonna, compound tincture of cardamom, and the strong tincture of ginger. So consider ginger. So ginger contains the characteristic pungent constituents. So a suitable solvent passes through the prepared ginger drug and extracts the soluble constituents here. So cardamom contains some aromatic constituents. So the extraction process helps transfer the suitable constituents into a liquid preparation. Okay, so always connect the crude drug with its chemical nature. Reserve percolation. In reserve percolation, the first portion of the percolate is reserved. So for example, the first 3/4 may be separated. So percolation is continued. So the remaining drug is exhausted. So the weaker later percolate is evaporated to prepare a soft extract here. So this is dissolved in the reserved percolate. So finally the volume is adjusted. So why reserve the first portion? So because the first percolate is usually rich in the active constituents. So that's why we will reserve the some percolate here. So principles of the reserve percolation. So the first percolate is more concentrated, so it contains a higher quantity of the extracted constituents, so therefore it is reserved without heating. So later weaker percolates can be concentrated by evaporation. So this protects the important constituents in the stronger portion from unnecessary heat exposure. Okay, so imagine preparing a fresh fruit juice. Would you repeatedly boil your best concentrated juice if it heat could damage its flavor? No. So you protect the valuable portion. So reserve percolate follows a similar logic approaches. Okay. Percolation. Modified percolation is designed to save the heat, time and ministrum. So in simple percolation, the drug to percolate ratio may be approximately 1:4. So in modified percolation, it may be reduced to around 1:3. So the objective is greater process efficiency here. Okay, so in pharmaceutical manufacturing, so efficiency matters. So if two methods provide the suitable extraction, but one consumes less solvent and time, the efficient method may be preferred here. So however, the product quality must never be compromised. Okay, modified percolation process. So let's follow the process. The drug undergoes imbibition first. It is macerated. A portion of the percolate is collected. So further maceration and collection steps are performed here. So the process is designed to obtain the required percolate using controlled quantities of the ministrum here. So this is fractional processing. So instead of uncontrolled continuous solvent uses, so the procedure manages the contact and the collection more efficiently. Continuous hot percolation. So continuous hot percolation is also called as Soxhlet extraction. So it is used when the constituents are not freely soluble or difficult to remove from the plant cell. So heat and a continuous extraction improves the process here. So here the same solvent participates in repeatedly extraction cycles. So this is the beauty of the Soxhlet extraction here. So imagine a worker repeatedly washing the same material with the fresh warm solvent. So the extraction becomes more efficient here. Now let's look at the apparatus. So Soxhlet apparatus parts. So the Soxhlet apparatus has three major sections. At the bottom is the flask containing the boiling solvent. In the middle is the Soxhlet extractor. It contains the drug thimble and siphon tube. Okay, at the top is the condenser. So the condenser cools solvent vapors. Remember, flask boils, condenser cools, and extractor extracts. Siphon returns. So this one sentence can help you remember the entire apparatus. Okay. Working of the Soxhlet apparatus. So first, the solvent boils in the flask. Solvent vapor rises through the side tubes. So the vapor reaches the condenser. The condenser cools the vapors into the liquids here. So the liquid falls onto the drug. So the solvent dissolves extractable constituents, so the extractor gradually fills. when the liquid reaches the siphon level, the solution flows back into the flask here. So then the cycle begin again. So boil, condense, extract, siphon. So again boil, condense, extract, siphon. So this repeated cycle is the heart of the Soxhlet extraction. Okay, Soxhlet extraction cycle. So look at this cycle visually. So first boiling, condensation, extraction, siphoning, then back to the boiling. So it is continuous loop. So a beautiful example of the solvent recycling within an extraction system is, so the drug repeatedly contacts the condenser solvent. Students, imagine a tiny automatic washing machine for crude drugs. So it is repeatedly supplies the solvent and removes the extracted constituents here. So this is a simple way to visualize the Soxhlet extraction. Okay, advantages of the Soxhlet. So Soxhlet extraction offers a repeated extraction cycles here. So the drug receives the solvent repeatedly, so the extraction can be efficient and the solvent can be recycled within the apparatus. So it is used for poorly soluble constituents. So the process can improve the effective extraction without manually replacing the solvent after every cycle here. Okay, so however, remember one important point, so heat is involved. Okay, so therefore Soxhlet is not suitable for all drugs, especially heat sensitive constituents. Okay, factors affecting choice of the extraction process. So now we reach an extremely important decision making section. So how do we choose an extraction method? So 6 major factors should be considered. Character of the drug, so that is the first factor. Second, therapeutic value, cost, stability, solvent, and the concentration of the final product. So think of yourself as a formulation scientist. So you cannot say, I like a Soxhlet, so I will use Soxhlet for every drug. Science does not work like that. Okay, so the process must match the drug and the product requirement here. Okay, so character of a drug, so that is the first factor you need to be considered. So for soft or readily permeable drugs, so maceration may be sufficient. For hard or tough drugs, percolation may be improve the extraction here. So knowledge of the drug is highly essential. So you imagine comparing a soft leaf with a hard piece of bark. So can the solvent enters both at the same rate? No. So the particle structure, tissue hardness, and the permeability influences the extraction here. So therefore the drug itself tell us which process may be appropriate here. Okay, therapeutic value and the cost, so that is the next factor. So for a drug with a high therapeutic importance, so maximum and a controlled extraction may be required here. So for costly drugs, we should minimize the loss of the valuable constituents. So for inexpensive materials, a simple economical process may be sufficient here. So imagine extracting a very expensive active constituent. Even a small loss can have a financial importance, no? So therefore extraction is not only a scientific decision, it is also an economical decision also. Okay, stability of the drug. So stability is a critical factor. So if the constituents are thermostable, hot extraction may be possible here. So if there are thermolabile substances, so heat should be avoided. So for heat sensitive constituents, maceration, suitable cold extraction methods may be selected. So here is a simple example. What happens to the ice cream when exposed to the heat? Its physical stability changes. Similarly, some drug constituents can degrade when exposed to the high temperatures. So therefore, before applying heat, always ask, so can my active constituent tolerate this temperature or not? Okay. Solvent. So the solvent is one of the most important factor in extraction. A good solvent should dissolve the desired constituents effectively here. So but selection also depends on the safety, boiling point, cost, and compatibilities. So the principle, so like dissolves like can help us understand the solvent selection here. So polar compounds generally show better affinity for polar solvents only. So non-polar constituents may require less polar solvents. So but pharmaceutical solvent selection must also consider the toxicity and the final product requirements here. So a solvent should extract what we want, not simply dissolves everything. Okay. Time availability. So time availability also influences the extraction process. So maceration may require several days. Percolation can provide controlled continuous extraction. Soxhlet uses repeated hot extraction cycles. So imagine you need a preparation urgently. So can you always wait 7 days? So maybe not. So but speed alone is not enough. So the selected process must still provide the acceptable extraction and maintain the drug stability here. So we balance the time, efficiency and the quality. Okay. Scale of operation. So now consider the scale of operation. So a method suitable for a small laboratory quantity may be ideal for the industrial production. So in a college laboratory we may handle a few grams of crude drugs, but in industry hundreds of kilograms may be processed. So equipment capacity, solvent recovery, safety, energy consumption, process control becomes extremely important here. So think about cooking 1 cup of rice and cooking rice for 10,000 people. So the principle is similar, but equipment and the process control are completely different. That is scale up. So concentration of the product and conclusion. So finally the required concentration of the product affects the process selection here. So diluted preparations may be prepared using maceration or percolation. Semi-concentrated preparations may require the double or triple macerations for concentrated products. So further concentration step may be required here. So what have we learned today? Means extraction is the separation of the medically active constituents using a suitable solvent. So maceration works mainly through soaking, diffusion and dissolution. Percolation involves the controlled passage of the menstruum through a packed drug column, and Soxhlet extraction uses repeated cycles of the boiling, concentration, extraction and siphoning. So and most importantly, there is no single best extraction method for every drug. So the best method depends on the character of the drug, therapeutic value, cost, stability, solvent, available time, scale of operations and desired concentration. So before we finished, answer one question. So if a drug contains a heat sensitive active constituent, so would you immediately select the Soxhlet extraction? The answer is no. You first consider a suitable cold extraction method. So that is the scientific decision making. So thank you so much for your time and attention. I hope this presentation helped you understand the extraction processes in a simple and a practical way. So keep exploring, keep learning, stay curious and stay inspired. If you found this session useful, please like, share and subscribe to Medi. Press the bell icon for more pharmacy learning videos. So thank you everyone. See you in the next session.